it's a little bit of an interesting topic. I hope we'll enjoy as we proceed with the presentation. So what we've been learning so far are more of the infectious microorganisms that we've been learning so far are a little bit different from prions. We've been talking about, I know you've gotten classes for bacteriology, you've done virology, which is this class, you've probably done mycology. And so those are infectious agents that you have learned so far. But prions is a little bit different. We will see how just a proteinaceous particle is able to cause an infection. And it's a little bit interesting. Prions, they behave like viruses, but they are not viruses. So we will learn about them. We will see their characteristics, how they cause diseases, where they cause diseases, and which diseases they cause in humans and in animals. So this is the outline of our presentation today. As I've said, we will learn about some characteristics of prions, what exactly they are, the transmission dynamics, how do they cause diseases in humans, and speak a little bit of animals, because also animals take part in a way for how prions cause diseases in humans, and then look at their clinical manifestations of the diseases they cause, look at the diagnosis, if there are any, and treatment or prevention, if there are any. So we'll begin by doing a comparison of prions and viruses, and just look at some of the similarities and the differences between the prions and viruses. So when you look at the feature of whether the particle contains a nucleic acid, whether it contains RNA or DNA, then we see that prions don't contain RNA, neither do they contain DNA, but conventional viruses do. So viruses are either RNA or DNA. And then when you look at whether the particle contains protein, and prions do contain protein, yes, and the proteins are cellularly encoded. So these proteins for the prions, they are encoded by the cells of maybe the human or the animals. While for viruses, they also contain proteins, yes, but the proteins are encoded on the viral chains. When you look at the techniques that can be used to either inactivate a virus or a microorganism, such as use of ultraviolet light or heat, or other mechanisms like use of formaldehyde, we see that prions are inactive to these agents, while viruses are killed by these agents. So when you look at the appearance in the microscope, the prions will appear filamentous or amyloid-like, while the definite shapes for viruses are icosahedral or helical. And because these prions are encoded by cellular genes, normally the body does not mount an immune response to them. So in this case, you will not see an antibody against prion in case a prion infects a human. But if a virus infects a human, then you can be able to detect antibodies against that specific virus. Similarly, you will not, with a prion infecting a human or an animal, there will not be induction of inflammation because there is no immune response being mounted. But for the case of viruses, there is an inflammation being mounted because of the presence of the foreign virus in the body. So these prionioses are generally called transmissible spongiform encephalopathies. That is the name for the diseases that these prions cause. And this slide shows which particular prionoses are for human, diseases and which ones are animal diseases. So we'll start with the human prionosis, which are a little bit, the words are a bit difficult to pronounce. So as we go ahead, you can use the short forms of the words, because some of these words are Africans, we may not be able to pronounce them very well. So just to give an example, we have the Creutzfeldt-Jakob disease, the CJD, and then we have the fetal familial insomnia, FFI, then the GSS, Kuru, and the variant CJD. So those are the human prionioses. While for animal prionisis, we have the mad cow disease, that is also known as the bovine spongiform encephalopathy. And we have the prion that causes disease in sheep, that is called scrapie. And the one that causes the disease in deers and mules, the disease is called chronic wasting disease. So those are just examples of some of these transmissible spongiform encephalopathies that are categorized based on which ones are in humans and which ones are in animals. So prionisis can be classified as either familial or hereditary. So those ones are genetic. They are transmitted genetically. And then we have the sporadic ones, which can occur due to point mutations in somatic cells. And then we have the ones that can be infectious. So they can be transmitted via probably ingestion of meat or beef from an animal that is infected. So those are the three classifications for the prionisis: the ones that can be infectious due to ingestion of meat, the ones that can be sporadic due to point mutations of the gene that encodes for the prions in the cells, or due to hereditary, that is, it's genetic. So just to give an understanding or a definition of what prions are. So prions are small proteinaceous infectious particles that are resistant to inactivation by procedures that usually modify or denature proteins. So as we have seen on the slides before, they are protein in nature. They are encoded by the cellular gene. So there's a gene on the cells that ideally just encode for the prion protein. And we have also seen that they don't have RNA, neither do they have DNA. And they cannot be inactivated by procedures that we usually use to, inactivate viruses, such as using of ultraviolet radiations, or using heat or boiling, or using agents such as formaldehyde. So these agents, those particular agents that usually denature proteins or usually kill viruses, cannot be able to inactivate these prions. So, but there are other ways that you can use to inactivate prions, such as use of phenol or ether, or use of hypochlorite that can be used to inactivate these prions. So this is just an example of telling us how different they are from other microorganisms, such as viruses. So then this may be a repetition, but it's good for us to understand.
Dr. Obiero, we've lost you.
Just for the short hiccup. Can you see the slides?
Yes.
All right, thank you. So this is where we stopped, but we had talked a little bit about some of the characteristics of prions. So we'll go through them again. As we said, they are made of protein. They are resistant to inactivation by the methods that we normally use to inactivate other microorganisms and viruses. And they reproduce by converting normal protein to abnormal protein. So we will see this in the next slides. These particular characteristics, how they the normal protein in the cell is converted to an abnormal protein, and how they recruit other proteins to be abnormal. And then they do not induce the immune system because they are cellular proteins. They are viewed as self. So they are tolerant. The immune system is tolerant to them. So because they are self, so the immune system will not be mounted against them. Then they cause vacuolation of neurons. So in this case, this is their pathogenesis, how they are pathogenic to the neurons. They cause vacuolation because once they recruit newer proteins to be abnormal, then that becomes neurotoxic to neurons and ends up killing those neurons. And this gives a sponge-like appearance of the brain. So we'll see that in the next few slides. So this was just to give you an overview of what the prions are and what are their characteristics. So this slide gives is for us to appreciate the difference between the normal prion protein and the abnormal prion protein. So on the left, we can see the normal folding pattern of the prion protein. So the normal prion protein is called PRPC, which means which just means prion protein cellular. Remember, we said these prion proteins, they have genes that are encoded in the cell. So normally the cells would produce these particular proteins. So the normal one is called PRPC, which is prion protein cellular, and the dominant form, or its dominant tertiary form when it is folding, is the alpha helices. You can see, I hope we know what alpha helices and what tertiary folding form for proteins are from biochemistry. So we can see here on the left, it has more of alpha helices and less of beta sheets. These are the beta sheets, and these are the alpha helices. So the normal prion protein, which is PRPC, prion protein cellular, has the dominant form as alpha helices and less of beta sheets. So when this prion protein becomes abnormal due to other mechanisms, either whether it's mutation or whether someone or an animal injects a meat or beef that is contaminated by prions, and the normal proteins becomes converted to the abnormal one. So you can see now on the right, we have the prion protein that is abnormal called PRPSC. So PRPSC just means prion protein scrapie. So that is the abnormal prion protein, and it's called prion protein scrapie because scrapie is a prionosis. We saw it in the sheep, and it was first identified in this abnormal prion protein was first identified in the sheep. So for the abnormal prion protein, we can see the dominant folding format is the beta pleated sheets and less of alpha helical. So I hope we can appreciate that. So we know that the function of a protein is determined during the folding, during the tertiary level. The protein, the polypeptide, can have the same sequence of amino acids, but once this particular polypeptide folds into the tertiary level, then the function of that protein can be different. So in this case, if the normal prion protein folds and we have more of the beta pleated sheets and less of the alpha helices that is in the normal protein, then this is a misfolded protein that now becomes pathogenic in the cell. I hope that makes sense. So this gives further explanation about alpha helices and beta chains. Just to concentrate more on beta chains, you remember we've said that the normal PRPC has more of alpha helices and less of beta pleated sheets. So if we look at the beta pleated sheets, normally the amino acids are in linear format. And these beta pleated chains, they have the tendency of, one beta pleated chain lying next to another, then another one on top of the other. So they form like, they easily attach to each other using non-covalent bonds. So this is also what we will see next, that once an abnormal prion protein can be able to attach to another prion protein and then recruit it and form aggregates. And that is like the pathogenesis of killing the neurons in the brain. So this also gives a further explanation of how folding of the proteins can make different proteins that have different functionalities. So we can see, for example, here, insulin has less of beta sheets and more of alpha helices. And we all know insulin functions differently from a ribonuclease, which has more of beta sheets and less of alpha helices. So we'll now get to try and see a difference between the normal cellular prion protein and the scrapie protein. So for example, in terms of structure, what we've just been saying is that the cellular protein, prion protein, PRPC, is predominantly alpha helical, while the scrapie is predominantly beta sheets. In terms of solubility, the PRPC is soluble, while the PRPSC is insoluble. And in terms of infectivity, the normal prion protein is non-infectious. It cannot infect other prion proteins in the cell. And while the scrapie is infectious, it's able to make other prion proteins change their conformation or misfold like it. And then another thing is that in terms of resistance to proteinase K, the cellular PRPC can be degraded by proteinase K in the cell, while the scrapie is resistant to degradation by the proteases. So I want us to take a minute to go through this question, and we'll probably write the answers on the charts, and then we'll continue. So one of the following is true about abnormal prion proteins. A, they strongly induce the host immune system, reproduce through budding, contain RNA but not DNA, cannot be inactivated by formaldehyde. So let's give our answers, and then in the next 2 minutes, we proceed. Okay, so I can see the answers streaming on, and most of us are getting it correct. So we'll proceed. All right, thank you for the answers. So what are the transmission dynamics for the prionosis? We already talked about it in some of the other slides, but we'll proceed and talk about it more. Number one, it can be due to sporadic changes in the somatic cells. So it can be due to point mutation in the gene that is encoding the prion protein, or it can be iatrogenic, meaning that it can arise from a medical context in terms of maybe someone getting blood transfusion from someone who had been infected by the prions before, or getting organ transplant, such as corneal transplant from someone who had a prionosis before, or because we've seen they are a bit resistant in terms of inactivation. Sometimes if we do not sterilize the medical equipment very well, that can be also a source for transmitting from one individual to another. And it can also be hereditary, whereby the gene encoding for the prion protein becomes mutates in the germ cells, or it can be through ingestion. That is, you can probably, if you ingest beef from a cow that has the BSE, that is the mad cow disease, or it can be through cannibalism. So there are some tribes that, like we'll see it as we proceed, but there are some tribes in New Guinea that used to practice cannibalism, in that it was like a ritual for that particular tribe to eat the brains of the dead. So in such a case where they eat the brain of someone who already had been infected by a prion, then that is a route for that transmission to the next person. So this gives an outline of the pathogenesis with prions. And the key feature is that is accumulation of the abnormal prion protein. As I said before, the prions, they are infectious, the scrapie, the PRPSC, they are infectious, and they have the tendency of recruiting other normal prion proteins to be abnormal. So once they keep on recruiting other prion proteins to be abnormal, and they cannot be degraded by the proteases in the cell, so then they form plaques in the cells. And if it is neurons, they form plaques in neurons, and these plaques can be neurotoxic to the neurons and end up making the neurons die. So from bullet one, the normal cellular protein PRPC is found on the surface of brain cells and other cells. So it's majorly found on the surface of brain cells. And infection or mutation of the PRNP gene that is usually located on the short arm of chromosome number 20 can cause PRPC to fold into PRP scrapie. So if there is either a mutation or an infection through maybe ingestion of the meat that is infected by this prion, then this can cause the normal prion protein to misfold into the scrapie type. And then this scrapie type will now dissociate from the cell membrane, because we've said they are surface proteins on the cell membrane, they dissociate. And then they cause more they cause more normal proteins to misfold. So they infect other normal prion proteins, and these other prion proteins will also misfold and dissociate from the cell surface. So then they accumulate and form proteinaceous plaques between the brain cells. And the aggregated forms are internalized into the cell. And these plaques that have been internalized, they are neurotoxic to the neurons. So the neurons will die. And then once the neurons die, there's a formation of vacuole, like where the neuron was, it's like a hole. And how the brain will look like, it will look like a sponge. That's why it's called a spongiform appearance. Or if you know how the Swiss cheese looks like, it looks like a sponge that has holes. So that's how the brain will look like. So this also is just a diagram illustrating the same thing that we have been talking about. We can see the normal PRPC anchored on the cell membrane. And once this abnormal one that has already formed an aggregate comes into contact with this normal one, then it converts it to the abnormal one and makes it misfold. And then once that happens, they form these aggregates that are here. And these aggregates are plaques on the in between the brain cells, which can be internalized inside the neuron. And these, once they are internalized, they become neurotoxic, and the neuron can die, and then forming the vacuoles, giving the appearance of the spongiform. So this is how the brain tissue will look like. This is on the extreme left is the spongiform appearance, whereby you can see the holes. And the normal brain has no holes or vacuoles. So just to give more description of some of these TSEs, the transmissible spongiform and encephalopathies that are in humans. So this slide just gives further description of the pathogenesis of this, and it has concentrated mostly in human. So we'll start with the Kuru, which is the disease. And this is an infectious form. It is transmissible. And the important feature about it is what I talked about before, is we had the Tori tribe in New Guinea that had the ritual of eating brains of the dead. And for those people who already had prions, that is how they would transfer this infectious protein to the next person through either handling of the brain tissue or that nature of cannibalism. And then the next one is the Creutzfeldt-Jakob disease, the CJD. And this one can be either infectious, or it can be hereditary, or it can be sporadic. So the infectious form one is gotten through iatrogenic means by corneal transplant. If someone gets a corneal transplant for someone who already was infected by CJD, then that prion can be transmitted to them, or through brain electrodes, or through growth hormone from the pituitary gland of someone who was already infected by this particular disease. So then the hereditary format is due to mutation in the germ cells, and the sporadic format is due to point mutations in somatic cells, and this is the most common one. Then we have the type that is called variant Creutzfeldt-Jakob disease. This one is transmissible or infectious, and it is hypothesized that it is usually acquired by eating meat or nervous tissue from animals with mad cow disease. So the animals that have, if the cow is already infected, and we said cooking or boiling will not kill the prion, the scrappy. So if you eat meat from animal that was already infected, then the humans will get the variant CJD. Then we have this Gutzmann-Straussler-Scheinker syndrome that is also hereditary, and it's due to mutation in germ cells. And then the last one is the fatal familial insomnia that is also hereditary and is due to mutation in germ cells. So these are some of the clinical features that someone who has TSC can experience. and most of them are neurological dysfunction features. For example, there is cognitive impairment. You can have ataxia or myoclonic jerks, mutism, and for fetal familial insomnia, you'll have insomnia, dysautonomia, and motor paralysis. And normally these features, they take a long incubation period. They are slow to develop. And at the end of the day, the disease becomes very fatal and the person ends up dying. So for diagnosis of this priognosis, most of them, it's at late stage when someone is already presenting with the clinical presentations that we've seen before in the previous slide. Some of those clinical presentations that are indicative of, neural dysfunction are what you will see and what someone might suspect to be a priognosis infection. And then we need to exclude differentials such as blood counts, because remember we said that this prion protein, they are not viewed as foreign by our blood cells. So in such a case, there will be normal levels of white blood cells, because we are not mounting any immune response to them, so that we cannot use that as a format for diagnosis. And then when there is some neurological dysfunction or in some diseases such as the priognosis, or diseases such as Parkinson's and Alzheimer's, these particular proteins are usually very important in their diagnosis, the 1433 protein and the tau protein. So this can help in diagnosis using the CSF analysis. Then you can also do an EEG, electroencephalogram, to see the brain activity. Of course, you expect that because the prions are dying, you'll definitely see the deficiency in brain activity. Then you can use MRI to visualize how the brain looks like. And brain biopsy, or during postmortem, then you can see the pathologic conditions that this priognosis has caused, and you'll see the spongiform formation in postmortem. So unfortunately, with priognosis, there is no treatment. There have been, research for medications that can be used, but as per now, there's no treatment. And prion diseases are fatal within months to years. The person, when they start showing the symptoms, the person will die. And the only way is to give palliative care. So what are some of the prevention strategies? Is to sterilize surgical equipment very well. So we can use sodium hypochlorite for those medical equipment that cannot be autoclaved. You can use the sodium hypochlorite. And then you can do screening of blood and organs, because it has also been shown that blood transfusion from those people who are infected by priognosis can cause this disease to be transmitted to other people. So this was majorly in people in Britain who had the variant CJD form. They were able to, some few people were able to transmit this infection via blood transfusion. And some of those people who migrated to, and it's more of region-based, because if it does not, I don't think in Africa or in Kenya, we do screen for priognosis for blood transfusion. But for regions where they have selectively experienced this, I think it's very paramount, because for those people who migrated to the United States, the United States government had put in prevention strategies that people who had come from either Britain or the UK, and were blood donors, then their blood had to be screened for this particular prognosis. Then ban on meat and bone meal in animal feed. So this is also an example of why people were getting the variant form of CJD in the UK is because animal feeds, they were feeding animals to animals. So for example, they can make cow feeds using animal bones, and then they feed it to the cows. So it's like you're feeding cows to cows. Or they could use the brain of the sheep to make feeds for the cows. So you see, in such a scenario, it's easy to transmit the prognosis from animal to animal. Then when human beings eat that animal, then they get the variant format of CJD. So the prevention strategy is do not feed cows to cows. And I would also say, do not feed sheep to cows. So this is our last question. Regarding prion-mediated diseases, which one of the following is the most accurate? A, prion-mediated diseases are characterized by vacuoles in the brain called spongiform changes. B, variant CJD is a disease of cattle caused by the ingestion of sheep brain mixed into cattle feed. C, Kuru is a prion-mediated disease for which the diagnosis can be confirmed in the laboratory by a fourfold or greater rise in antibody titer. And D, in CJD, only neurons latently infected by JC virus produce the prion filaments that disrupt neuronal function. So let's give our answers, and then I'll take questions. So the answers are coming in, and I can see we have really understood what prionoses really are. So maybe I can now take questions before we leave. If you have any question, please write it on the chat so that we can discuss. So if a person gets tested and finds that they have a low concentration of these prions, but no symptoms, can something be done to remove those prions, or at least halt their influence over normal proteins? So unfortunately, by the time you'd be, and most of the time when diagnosis is being done, by the time you'd be getting that someone has a prior nose infection or has TSC, it will be mostly at the last stage when they're already exhibiting neural dysfunction symptoms. And at that stage, there is ideally nothing you can do. That's why for that stage, the treatment options is just palliative care. So I hope I've answered. Does it make sense, Kelsey?
Yes, thank you.
All right. Okay, let me see the next question. A goat that eats meat instead of grass is a dog. Is a dog, Argentina proverb. All right. That's interesting. So do these abnormal prion proteins cause granulomas in the brain of the affected person? So they don't cause granulomas, they are just neurotoxic. You see, once they form those aggregates, and these aggregates cannot be digested, or they cannot be broken down by the cellular proteases. Then once they are internalized into the cell, into the neuron, it becomes neurotoxic to the neuron. So the neuron just dies. So it doesn't cause granuloma, the neuron dies. And in the brain, when the neuron is dying, we also have microglial cells, right? That are always just, hovering around to see what they can engulf. So when they see these neurons that are dying, they engulf the neuron and it leaves that hole where now we see the vacuoles. So it's just an indication of dead neurons. And that's why as more neurons are dying, then we are able to see those neurological dysfunctions. So can a human really survive without these prions? Yeah, these prions, they are encoded in our cells, and it's hypothesized that they have a function in signal transduction at the synapses of the neuron. So it's not really clear what their specific function is. That's just an hypothesis. But since it's a gene that is in the cells and it's producing proteins, it's not like a silent gene, it must be having a function. So fertility of prions is 100%. I will say so. So Doc, you said prions are hereditary. So we saw the different formats here, the different ways that the prions can be transmitted. We saw it can be through infectious, when you ingest an infected, meat. Number two, it can be either sporadic, so that is when there is point mutations in the somatic cells of the gene that is encoding for the prion protein. And it can also be hereditary. So when there is a mutation in the germ cells, then it becomes hereditary in that it can be, transferred from one generation to another. So those are the explanation. It's not only hereditary, it can be due to point mutations or sporadic mutations, or it can be due to ingesting contaminated meat products. So what's the purpose of this PRPC in the brain? So that's what I said before. Their purpose is not very clear, but it's being said that they have a function in signal transduction at the synapses of the neurons. So we said they don't get inactivated by formalin and other chemicals that inactivate proteins. Yes, but they can be chemicals like formaldehyde or formalin, radiation such as the UV that we use sometimes in molecular labs to inactivate viruses, and just generally heating or boiling to normal temperatures, those ones will not inactivate these prions, this crepey form. But other methods like using sodium hypochlorite can, and if you use phenol or ether, they can also disrupt or inactivate this prions. So that's why it's important to use, when you're sterilizing the medical equipment, it's important to use sodium hypochlorite for those equipment that cannot be autoclaved. So do we have prions? Yes, we do. We have the gene that encodes for the prion protein, but it's the normal, I want to believe it's the normal prion that we have, not the abnormal, because when you get the abnormal, it's slowly progressing. So you'll just know once you start developing the symptoms of neurological dysfunction. So Kelsey, that is where gene therapy comes in, though it's highly experimental, Sai. Okay. How are prions neurotoxic? So as I said before, in their pathogenesis, you remember they keep on, so the scrapie format keeps on recruiting the normal ones to be the misfolded ones. And remember, these misfolded ones, they form aggregates that form plaques and in between neurons in the brain. So they form those plaques. These plaques, remember, they are insoluble, so they can't be degraded by proteinases. They are also, yeah, they are insoluble, so they can't be degraded. So the only thing that happens is that the neuron ingests it, like takes it in. So once it's taking in these plaques, it just disrupts the normal function of the neuron, and the neuron dies. So that's why I'm saying it's neurotoxic, because those plaques are being ingested, the aggregates are being ingested inside the neuron. It's disrupting the normal function of the neuron, and the neuron dies. So what is zombie deer disease? I would say it's the chronic wasting disease that we just saw, and it's a Prions, because remember, these Prionoses, when they infect the brain and your neuron cells are dying, you have a neurological dysfunction. So that's why I don't know the name of the zombie deer disease, but I will describe it as the chronic wasting that affects the deers and the mules. And you can also see the behavior of the mad cows, because their neurological function is not there, or it's degenerating. So if you can relate it to the mad cow disease, whereby the cow is very aggressive, and there is those symptoms of neurological dysfunctions being displayed, then maybe, I don't know if it's a scientific term though. We may need to find out. All right. Is it safe to say prions are the most dangerous living things to humans? Maybe we can get a general comment from the class on that. Do you think it's them? Because it's very rare. It's like, for example, the VJD one happens in 1 to how many millions of people. So maybe it's very fatal, but it's not so common. So is this disease caused by cows only? Can camels cause TSAs? So it has been demonstrated in cows so far. We've not seen it in camels, or I have not come across, articles or papers talking about TSAs in camels. So yeah, if you have come across, then we get new knowledge every day. So chronic wasting disease, the TSC version of deer and elk. Okay, someone says don't justify nyama ya ngamia. All right. So what's the incubation period of prion disease? So most of the time, it has a very long incubation period. I can't put it into specific years, but most of the people who from the articles that I have come across, they say that the CJD will be demonstrated in way older people. So if you the mutation happened when you're younger, or if you ate meat that is contaminated when you're younger, then you get the variant form when you're a little bit way older, around 70 years. So you can see the incubation period is very long. Kindly elaborate on sporadic. So when I say sporadic, what I mean is it's just point mutations that happen on normal somatic cells. We have somatic cells, and then we have germ cells. So if a mutation of the gene encoding for the prion protein happens on the germ cells, then it becomes hereditary in that it can be transferred from one person to another. But if the mutation appears on the normal somatic cells, then it's the sporadic format, and this one cannot be transmitted. You cannot transmit to another generation. It's just in that particular person. I hope that makes sense. All right, so Kelsey says months to years, I think. Yeah, we can't really put a definite number on it, but we know it's a very long incubation period. But for months, it will be months to months, I will say, for someone to, once you start displaying the characteristics of neural dysfunction, within months, someone can die. But for years is the incubation period that it will take for these particular clinical manifestations to be shown. So do the abnormal proteins present many years after infection? Do the abnormal prions present many years after infection? I'm thinking you're trying to ask about the long incubation period. But remember, when the abnormal protein, let's say if the abnormal protein infects right now, it may take time for the clinical manifestations to portray, but it will keep on infecting other prion proteins. I don't know if I'm making sense. It will keep on making other proteins misfold and slowly by slowly killing the neurons. So so it will be slowly progressing, and then as when the clinical symptoms now start to present, is because now most parts of the brain has been damaged. So it has long incubation period, yes, 5 years or more. Does mutation of gene encoding prions occur only in animal or both animal and man? It occurs in both, because once you have a form of, for example, if we talk about the CJD, the one that is not variant, leave alone the variant one. So that particular one, the sporadic mutations will occur in man. Then the variant format will occur if man now eats meat that is contaminated. I hope that makes sense. So it can either be a mutation that just has taken place in man, and now the priognosis will be manifested, or it can be, the variant format can be, the man went and ate meat or beef that is from an animal that was already infected, and then now they get the variant format. But for the animal also, there must have been mutation for that abnormal protein to occur. So can touching brains in the anatomy lab without gloves transmit prions? A demonstrator once told us it can. Yes, that's why we say when you are in the lab, put on the required PPEs, because, for example, in the case of the Kuru disease, it is also hypothesized that apart from that cannibalism act of eating the brain, also the act of just, opening the skull to take out the brain may have been a way to transmit, these particular prions to humans. So always be safe in the lab, put on the proper PPE that is required. Physiological prion protein is found on the surface of cortical neurons and has a protective mechanism against free radicals. Conformation change is triggered via misfolded. So there are very many, hypotheses on the functions of the normal prion protein. So yeah, you can read across that, but there is no definitive way of saying that this is the function of, the normal prion protein. But we agree, once a conformational change occurs and it gets misfolded, then it can recruit other normal prion proteins. The cadavers are well treated. They just don't bring anybody to the lab. I don't understand what Bunyavirida is trying to say, but remember, even if the cadavers have been treated by, they have been fixed by maybe formalin or whatnot, remember what we said, formaldehyde does not inactivate prion. So if you don't keep safe and put on the right PPE, you can be able, if that particular cadaver was had a prionose, it can easily infect you. If it is hereditary, does the baby grow to become an adult? Remember, we said it has a long incubation period and it is slow progressive. So they will grow and the clinical manifestations might start being shown maybe when they are adults or when they are older. Someone says incubation period can be 5 to 40 years. We said that chaperones are responsible for normal protein folding, right? Now, is there any relationship between peers and chaperones? So, yes, chaperones are responsible for normal protein folding. And in a normal scenario, if a protein does not fold well, the chaperone will either make it fold the right way, or this protein can be, now if it still misfolded, then the nucleases or the proteinases can go ahead and degrade this particular protein. But that's not the case with PRP scrapie. And remember, PRP scrapie are insoluble, and even once they misfold, the chaperone is not able to make it fold right. And as it bypasses that stage for the chaperone, then the next stage is, to be degraded by the cellular proteases. And remember, the scrapie format cannot be degraded. So that's why it keeps on, progressing and forming aggregates. So the normal prions, what are their functions in the body? As we said, it's still a little bit unclear, and there are many people coming up with so many hypotheses. And one that I mentioned is they may be involved in signal transduction at the synapses of the neurons, but you can read more about it. There is no definite, function that has been identified. Are these things proteins even? Yes, they are proteins because there's a gene in the cell that encodes for them. And what do our genes do? They encode for proteins, right? And we even have its polypeptide format, and we can see that the normal format has more of the alpha helices and less of the beta pleated sheets, and the abnormal format has more of the beta pleated sheets. So yeah, they are proteins. So it does not cause granulomas, it causes plaques, and plaques are just aggregates of the abnormal scrapie format that cannot be degraded in the cell. So because they can't be degraded, they are internalized by the cell, and they become, they disrupt the function of the cell, and the cell dies. So what are the functions of PRPC? I think I answered that. Can eating just one piece of the brain of the cadavers in the anatomy lab cause TSA asking for a friend? I don't know why you'd want to be eating the brain of a cadaver, but we've seen from Kuru that this can still cause, because once the abnormal format comes into contact with the normal protein, it converts it. And remember, it becomes now infectious. It keeps on converting others slowly by slowly. So there you get the answer. Are PRPCs found in the brain only, or can we find some in the, so it's not only in the brain, it's also found in other cells, because they are in the membrane of the cells. They are transmembrane. So these particular proteins can dominantly in the brain, but they can also be found on other cells. In an anatomic pathology, we are told that unfolded protein accumulation in a cell can mediate apoptotic cell death. Does the prion activate apoptosis in the long run? I will say from the pathogenesis, I will say accumulation of these aggregates. What makes the cell die is the accumulation of these aggregates that are internalized, disrupt the normal function of the cell. So once they disrupt this normal function, then the cell dies. When the cell dies, the microglial cells that are usually like macrophages on the brain, they come and they find this cell that has died, they engulf it, they like eat it, and then there is a hole that is left that looks like the vacuole, and that gives the look, the spongiform look. But in a normal scenario that we said, unfolded protein can either be, killed, they can make the cell die. But remember, this is not a normal scenario, because this prion proteins, scrappy, are a little bit resistant. Though, if there is programmed cell death because of the presence of the aggregates inside the cell, then that would make sense. But if it is, trying to rectify that unfolded protein, then that cannot happen. So unless it is just killing the cell because there is already aggregates of so many unfolded proteins, then that will make sense. So, okay, do prions cause granulomas? I think I answered this question. It's not a granuloma, it's just plaques that gets internalized and disrupt the function of the cell. Is using sodium hypochlorite not conventional? Yeah, in the lab, the sodium hypochlorite is supposed to be used depending on which type of lab you're working in. But normally, for, like, let's say, sterilization of medical equipment, the first thing that probably comes into mind is autoclaving or such kind of methods. Autoclaving also works for for prions. They can inactivate the prions, but in a case where those medical equipment cannot be autoclaved, then you can use sodium hypochlorite to inactivate. So yeah, sodium hypochlorite is used in the lab. It's usually around 5 to 10% concentration for decontamination in the lab. How does autoclaving at 134 denature the prion protein? And we said heat cannot denature them. So we said heat, the normal boiling heat, you see the normal heat that you use for, let's say, cooking food, is not as high as the heat that is in the autoclave. So that's why I said normal boiling temperature or normal heat that you use for cooking cannot inactivate them, but autoclaving can. Some can be found in CSF. So when heat and pressure is used, it denatures the protein. So that's what Songok says. and Benson is giving the appropriate temperature. And you should also check the pressure that is involved in autoclaving and the time. So the temperature, the pressure, and the time, it's a combination. It's not only the temperature. And remember, there are different methods of sterilization. Because you can either have, heat and pressure, or you can just have dry heat, or so you can read through that too. That is extreme heat. Let's say you do it for 1 hour, that will be enough to disrupt the entire secondary structure of the prion. What was the incubation for infections? So I'm trying to understand Yvonne's question, but I think she means how long does it take once someone is infected to the time the clinical symptoms start manifesting. We've said it's a very long incubation period, but it's a range. Some have looked up and some have said 5 to 40 years. Some is beyond 40 years. So it's a long range that we cannot really put a definite number of years to it. So in anatomic pathology, we are told that unfolding protein accumulation in a cell can mediate. Does the prion? I think we talked about that. We answered that. And then someone has given a link to the chronic wasting disease for the zombie. Smart cannibals don't eat brains. Comrades spread wisdom. Yes. Okay. So I think when prions are misfolded, they cannot perform their function properly. And one of their functions, scavenging reactive oxygen species, they may lead to apoptosis, which may be mitochondrial, induced due to release of cytochrome C, activation of caspasesis. So yeah, protein misfolding will, in the long run. Yeah, if you have enough information backing this up, it might also be a way that the pathogenesis works, but the main pathogenesis is what I have described. But we can have more and more, ways, because it's not only one way that will lead to the death of the cell eventually. All right, I disagree. I think the neurotoxic effect of peers is direct. So Kamunya says the neurotoxic effect is direct. Okay, hi doctor. If we get prions transmitted through blood transfusion, will the body recognize those as foreign, as they are not of our own, or immune response still not be mediated? So remember we said the gene for the prion protein is like it encodes for that specific protein in the cells. So the only thing that the body can see as foreign is what is usually not encoded as genes in that particular. So for example, if there is a specific sequence for that particular gene, and the polypeptide that will be formed and the amino acids that will be formed are exactly the same, so we don't really have any foreign substance or an antigen that can mediate an immune response. The reason as to why you mediate an immune response is the body sees something that it doesn't normally have. But in this case, the gene is encoded in the cells, the polypeptide chain with the amino acid sequence same. The only difference is that in tertiary level, the folding is different. So that might not be enough to mediate an immune response. I hope that makes sense. Though PRPC scavengers, I don't think they're the main effectors of this role. Again, in mice where the gene for this protein was knocked out, They were normal and even showed reduced susceptibility to TSC. So the knockout mice that Kamunya is talking about, they have the gene for the prion proteins, but it's not active. So the gene is like a silent gene, and they are not producing prion proteins. So in those knockout mice, there's no prion proteins being produced. So even if you inject them with a prion protein, then there will be no pathogenesis, because it needs another normal prion protein to convert it to the abnormal one, and then it continues, the system continues like that. So if the gene, like in that particular one, the gene is silent and it's not producing the prion proteins to be converted, then there is no pathogenesis. So what was HSP60 leading to normal folding? So Harrison, please elaborate on your question. Just explain what you mean by HSP60. Yeah. What will be the effect of aggregation of on those other cells? So as I said, the pathogenesis is when the abnormal prion protein makes the other normal prion proteins to be abnormal, then it aggregates and it's internalized in the cells, and this makes the neurons dysfunction, or it alters the normal function of the neurons. Then that particular one is killed. So remember, these prion proteins are on the cell membrane. That particular one is killed. Then it goes to another cell that is also expressing this particular prion protein. It's also changed. The cell is killed. So we have so many cells being killed, showing in the parenchyma of the brain. You'll see an indicative of holes. It's like they are being infectious, like they're acting like viruses. You see, once a virus infects a cell, if its mechanism of pathogenesis is lysis, it lyses that cell. The next one is also like, so that is how it keeps on infecting other cells. Don't think there's significant damage because the pairs can be found in other tissues, but their pathological changes only occur in the brain. So, all right. Frank, you can't kill these things, not without ruining the meat. Okay, so are we satisfied with priognosis? We can read more about them. Some of these things that we are talking about are a bit speculative, and they're based on maybe data that has been shown in places that these priognoses have been affecting people. So you can read more about the same and get more knowledge on the same. So are we okay? Can we call it a day?
Yes.
All right. So could we say priognoses constitute emerging diseases? Unless you say, because emerging is something new, right? So I wouldn't really call it emerging because it has been there from way back earlier, because the very first prionos infections were, gotten a little bit way. So unless it emerges again in a population, then we can maybe call it a re-emerging But with some of the prevention strategies that have been put, and with it being a little bit rare, I wouldn't call it emerging. All right. Unless someone has another opinion on the same. Okay. So Kelsey gave me a go ahead to call it a day. So I guess we'll meet next week. So enjoy your lunch.
Thank you.